Hey, the Biochemistry Academy here. We began a serial course on enzyme, and in today's video, we'll talk about coenzyme and cofactors, understanding their properties and also the example of these coenzyme. Okay, let's pay close attention because it is a little tricky. Some enzymes do need non-protein components to carry out biochemical reaction occurring in the cell. These non-protein components are called cofactors. So an enzyme with its cofactor is called an holoenzyme. And if this enzyme loses its cofactor, the enzyme is called an apoenzyme. So an apoenzyme plus its cofactor is called holoenzyme. Now, the apoenzyme is usually non-active and it needs its cofactor to carry out its function. Let's give an example to understand this. And let's use the enzyme DNA polymerase. Now, the DNA polymerase need an ion, which is a magnesium ion, to carry out its function. So the DNA polymerase with the magnesium ion is called an holoenzyme, while the DNA polymerase without the magnesium ion is called an apoenzyme. It is the same with RNA polymerase that needs its sigma factor for its function. So let's continue. This cofactor can either be organic or inorganic. Now, in the inorganic cofactor, which are usually metal ions, the metal ions that are tightly bound with the enzyme are called metalloenzyme. But if they are loosely bound, they are called metal activated enzyme. Next, in the organic cofactor, it can be tightly bound with the enzyme or released freely after the reaction is complete. Now, if it is tightly bound, it is called a prosthetic group, whereas the organic cofactor that is released after the catalysis is called a coenzyme. Most of the coenzyme cannot be synthesized by the body, so they are derived from vitamins. Example of these coenzymes are NADH, NADPH, FAD, FMN, and etc. Now, do you understand? Okay, let's continue. Now we are going to understand the properties of this coenzyme in details. As we know, holoenzyme is made up of apoenzyme and its cofactor. And this cofactor, if it is organic, it is called coenzyme. Now, from the diagram, we can see the apoenzyme. And this apoenzyme, it is the protein part of this holoenzyme, which is responsible for the 3D infrastructure for chemical reaction. And the coenzyme, it is the non-protein part, but organic, which is required for optimum activity of the enzyme. Therefore, apoenzyme plus coenzyme equals holoenzyme. Now, one can define a coenzyme as a low molecular weight organic substance, which is heat stable. Now, this coenzyme is essential for biological activity of the enzyme. After the completion of the reaction, the coenzyme is released from the apoenzyme and can bind to another enzyme molecule. Remember, we said that most of the coenzymes are derived from vitamins, and these vitamins are vitamin B complex vitamins. Now, it has no role in determining the substrate specificity of the enzyme. It is the Apple enzyme that is responsible for the substrate specificity. Hey, so let us just continue. Let us see the various coenzymes that are derived from the various vitamin B complex vitamins. So the vitamin B1, also known as thymine, the coenzyme is thymine pyrophosphate, which is required for oxidative decarboxylation and transketolase reaction. Then the vitamin B2, which is also called riboflavin. Now the coenzymes are FAD and FMN, which is required for oxidation and reduction reaction. Then the vitamin B3, which is also called niacin. The coenzymes are NAD plus and NADP, which is required for oxidation and reduction reaction. Then the next vitamin B complex vitamin is vitamin B5, also called pantothenic acid. The coenzyme is coenzyme A or CoA, and it is an acyl carrier. Then vitamin B6, called pyridoxine. The coenzyme is pyridoxal phosphate, and it is required for transamination, deamination, and decarboxylation of amino acids. Then the vitamin B7, also called biotin. The coenzyme is biocytin, which is required for carboxylation reaction. Then the next vitamin B complex vitamin is vitamin B9, also called folic acid. Now, the coenzyme is tetrahydrofolate, which is a carrier for one carbon group. Then finally, vitamin B12, also called cyanocobalamin. The coenzyme is methylcobalamin, which is required for the carrier of a methyl group. So these are the vitamin B complex, and these are the coenzymes that are derived from these vitamins. There are other coenzymes that are derived from non-vitamins, and they include lipoids, tetrahydrobiopterin, and coenzyme Q. If this video have helped you, please don't forget to like, share, and subscribe to the Biochemistry Academy for more content like this. Arigato.